settle, they're able to join the coral reef ecosystems. And so this distribution and end location is influenced by their own biology, so their own life history, but also what the ocean's doing. And then when the ocean currents are moving around and they finally interact with the coastline again, sort of what the coastline looks like, and if it has big rocks and ledges, or if it's a flat reef, the settlement will act a little bit differently.
Quick interruption.
Oh,
Aloha.
thank you so much. All right. Yes.
Now we know the speakers.
Yes. So a little bit. So when we talk about coral reproduction, does anyone know sort of a method or two that corals use to reproduce? Spawning. Yeah, exactly. So you have corals that need to have their genetic material mate with another genetic material of another coral. And the way they do this is by releasing it out in these big mass spawnings. Has anyone seen videos where they have all these little particles and genetic materials going up, and then they mix, and then they join these little egg sacs, and then they start to travel around in the ocean? Another way is called brooding, where the actual coral itself is able to self-fertilize, and then they release a planulite already formed, ready to go, and that travels in around the ocean. So settlement and recruitment are slightly different things. So settlement is that transition from that planktonic larva into a coral polyp, and recruitment has to do with that process, but also the growth and of this coral and having it join the coral population. So you may see settlement and recruitment kind of used interchangeably through this talk. This study, we aren't monitoring the growth and survival of these polyps. So when we talk about settlement or a recruit, we're talking about a freshly settled coral polyp, rather than making comments on if it survives, how long it survives, and that type of thing. So what we're looking at is we're trying to see how many corals are settling or joining the populations in different areas along the coast, and also how this changes over time. So are there specific parts of the island that get more new baby corals than others? And is it consistent through time, or do we have peaks and troughs in the amount of babies that get delivered? And then what implication does this have for coral management for the future of our reefs? So that's what we're looking at. So our methods. Our study area really is all along the western Hawaii. It's a very coarse, so it's a very broad look at settlement. So we're able to talk about some stuff about a large area. So we have sites all the way from Waiakilio up north, all the way down south to Manuka. So that's a lot of coastline, and quite a few sites in and up between. So we have 3 in the northern region. Let's see, does this work? Oh, yeah. So 3 in the northern region, 3 in the middle region here, and then 3 in this sort of southern region. And the way that we measure this is by using settlement tiles. And so this is a settlement tile array. This is what we put out, and this is what we use to collect data. So this is on top. That is a terracotta pavement tile, like you would put down in your driveway, garden, what have you. They found that there's a really good surface and that corals like to settle on it. So it's really good to be able to use to measure how many corals are settling. And so they have a stainless steel rod, which is epoxied and secured into the reef. So these arrays, if everything goes well, they're going to stay in the same place. Now, there's 2 reasons why we'd have to replace an array and put it in a different location. One is the good reason is because it gets overgrown by coral. This is great. This is a good problem. We like this. But the other reason is if the surface or the location that it was attached to breaks through storm action, through waves, what have you. And so these tiles are put into the field, and at each of those 9 sites, we have 8 tiles. And so right here on the left, those are Ross Martin's hands. And he's replacing those tiles. We replace the tiles every 6 months. So what we do is we take off the nut and locking washer, we pull up the old tile, we put it in a Ziploc bag, we take it with us, and we put down a new settlement tile in replacement. And that's how we were continually monitoring for all these years. Now, one of the really important things about this design is there's a little spacer here. And what that does is it creates this environment. And the shallower waters, the corals like to settle in a more protected area because with the waves and the swell, it's a little bit more turbulent. And this gives them a little bit of relief. So the most common places that we're going to be finding corals are on this side here. And then if you took your fingers and reached underneath the tile, that area right there underneath is also the most common place that you're going to find these corals. Once we pull them up, we soak them in a bleach solution, 10% bleach solution for 24 hours. We use bleach. It helps to preserve the calcium carbonate skeletons, but it helps to take away all the organic material. So it allows us to store them, and it also makes them not very stinky. Once then we're dried, completely dry, then we wrap them in bubble wrap and save them in plastic bags until it's time for us to look at them under the microscope. So a lot of these things are very, very small. So a lot of these coral polyps, we're going to end up being about 0.5 millimeter. And so using that microscope, we have to scan up and down to be able to find them in all sorts of nooks and crannies. Once we find one, we'll measure it, we'll identify it to the genus level. And then there's other information, the location where it was, if it's broken or damaged, and things like that. So this is a diagram of a coral skeleton. So we're used to seeing the big three-dimensional structures or the big mounds where there's hundreds of thousands of polyps. But for these, we're looking at just the one little corallite, so the one little tube that the coral polyps live in. So important things that I look for identification is the columella. So if it's present, it's going to be this big calcium carbonate sort of column in the middle. And so that's one of the things that's really helpful for identifying them. And then the other part is the structure of the septa. So these lines, these calcium carbonate structures that go in. Now, don't worry, I'll show you exactly what they look like. So looking at the genus Parietes. So do these start to look familiar? So the lobe coral and the finger coral, these are really, really common around our reefs. So this is what they look like as a single polyp. So identifying feature is, if you look at the corallite here, it looks kind of like a snowflake, right? And so this is what we use to identify these in the genus Parietes. And so they're a little bit smaller. On average, they're about a little over 0.5 millimeter. So they're one of the smaller ones. Then we have the genus Montipora. So rice corals, mainly kind of like crusting here. If you ever done some dives on Oahu, you kind of see a few more branching, or certain places on the coast, you'll see some branching. But for a lot of what I've seen in our dives, it's a lot of crusting. And they kind of look like a mess a little bit, right? It's a little bit like the corallite isn't as well developed, and the septa are they're not consistent. And so they're a little bit more jagged, and they just look a little bit more rough. But that's how they develop, and that's how they go. And then we have Hauselopora. So we're looking at the cauliflower coral, the grandis, previously Adua, but the antler coral. And this is one of the ones which has a nice defined columella in the middle. And these ones are a little bit bigger too. They're almost a full millimeter on average. So some of these ones, if you look really carefully, sometimes you can see them on the tile. You can't really tell what it is, but you can see that there's something there. And so as they start to get older, if we put a tile down and then a coral settles immediately after, then sometimes they can grow to multiple polyps. You can see the 3 different most common genera that we find on our coast. This is what they start to look like as they start to grow bigger and they start to make their larger colonies. So limitations of the study design, because we're replacing the tiles every 6 months, the corallite, they're very small. So we're only able to identify to the genus level. We can't do specific species. So we're not able to comment on which species of Porites, for instance, or if it's specifically Poecilope meandrina, the cauliflower coral, that's reproducing at these different sites. So we're just not able to do that. And then the resolution. So pretty much because it's a large area and we have only 9 sites covering this very large area, we're able to have some information about a large area, but we're not able to provide fine scale differences between habitats and locations in certain areas. There's actually a study that Dr. Rachel Carlton started during her work with ASU, and they're looking at, and they have a very fine scale design, which allows them to start to talk about like the differences in these reef habitats and how that affects settlement. So look forward to some pretty cool stuff coming out of that. All right, so analysis approach. So it is our duty to disprove that there is any difference between the sites and the years and across years and sites. So our alternative hypothesis, so what we're hoping for, if everything goes well, is that we're going to find that there is statistically significant, the stats are going to show that there's differences in settlement between the years or between the sites, and then between the sites across time. It is noted that while we do exchange our tiles every 6 months, we're only using the summer rounds because most of our corals here, they have a spawning season or a reproductive season where late spring to summer is when most of the corals will spawn. And so 78% of all settlement that we observed in this study, so almost 80%, occurred during the summer rounds. So that's why we ended up using just the summer rounds. Now, when you want to run and do math and statistics, you want your data to look really nice. These curves are real good. If you see our data, it does not look like that. And this is one of the unfortunate aspects of settlement data, is there's a lot of zeros and a lot of values that are very similar with many outliers. So some of the things you can do, you can start to work, you can start to do things called transformations to kind of force your data to fit this normal curve. But when we did that for ours, it just did not work. It didn't fix anything. So that meant we have to get a little bit more complicated. Now, for those of you who are interested in hearing about the model, we can do that. For the rest of you who are not interested, there's a picture of Pedro Pascal. So we ended up doing a generalized linear mixed model. And the reason why this was really well suited for our data is that it likes non-normal data, it works with skewed data, and it works with the presence of zeros in the data. All the things we had, and so we were able to run the analysis, and theoretically, we might have some results. So what did we find? So average settlement densities. So let's look at the coastline as a whole. So for West Hawaii, we found that on average, the settlement density was about 21.8 individuals per meter squared. So as a reference, that's what the average of the coastline is. If we look at the 2 northern sites, Waikilio and Puako, they have some very high numbers compared to the rest of the coastline. So they're over twice the average of the coastline. So that's something that we found really interesting. The lowest value is Keauhou at only 5.4 individuals per meter squared. So Keauhou is very low. And our site at Manuka was also relatively low, which for some of you who dove that area and are familiar with that area, Manuka looks really nice. And so there's something to look into there where they have low recruitment, but their area looks really nice. So another aspect of settlement, which is a little bit frustrating at times, is if you look at the standard deviation and the range, those numbers are relatively large. It means that the values could either be very small or very big, and that's not out of the ordinary, which What it means is that settlement is episodic. So we're not getting consistent values all the time. Depending on the year, different sites may have really high values and really low values, and that's normal. And that's something that we can expect going forward. And so what we want to do is we want to start to look at trends over time. So in all of this up and down of episodic nature of these settlement events, sometimes we get more, sometimes we get less. Over the period of our study, does it look like we're getting more recruits in areas, less recruits? Overall, it looks, so for the coastline as a whole, it may look like there's a slight negative trend, which there is, but if you look at all of the dots, they're not very close to the line, which means that that line is not really able to explain too much of that variation. It's not that great of a fit. And so this is a loss smooth line. So what it does is it reacts a little bit more to the changes in data over time. Normally the lines are going to be straight in one direction or not. This one, you can kind of see that it goes up and down a little bit. And that's the nature of our settlement, is some years will be good, some years will be bad, and some years will be right in the middle. We're going to get used to this graph because we're going to see this a few times, because we can look up here. We have some very abnormally high settlement for these years. So for whatever reason, one more time?
2015?
Yes. So 2014, 2015, right?
When the water was 85 degrees?
Yes. Which is one of those things where you're like, huh, that doesn't, like you think that it might be a little bit lower. But yeah, it recorded a little bit higher. We're going to get into that in a sec. And then also 2009. So right here, so this is the settlement densities of our site. So these are the values that we are seeing for each of our sites. The top three are in the north region, the middle, and the south. So on the left-hand side, you can see those scales. The scales are different for each site. As you can see, some of the sites up here in the top left, so Waiakilio and our site at Puako, they had some extremely high settlement events. So more than about 4 times, 5 times their average. And if you look, that's at around 250 individuals per meter squared. And at some of the sites like Kaupalehu, they never had a year that was higher than 30 individuals per meter squared. So there's something going on with these sites and something going on those years that cause them to have abnormally high settlement.
Quick question.
Yeah, for sure.
These data, 6 places per site?
8.
8. So n is equal to 8 for each one of those dots.
Correct.
Okay, thank you.
Yes, so each one of these dots is the average of all 8 tiles per site. And so you can see I have just the straight regression line and then also the loss smooth curve, or line, excuse me, so that you can see the difference between the general trend and how the years, the different settlement amounts through the years change. So if you look at Puako, pretty straight, pretty straight. They kind of look very similar. But if we look up here at Kaupalehu, you can see that there's an, it's not significant, but there is a negative trend. But if you look here, positive, 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 it hits about 2017. And then following 2017, it looks like there's a gradual decline. Opposite of that, Ke'eho. Who'd have thunk? Ke'eho is right about 2014, 2015, started to have an increase in settlement, right? Which, again, I know 2015 was a huge bleaching year for us. And 2014 was also a very large year for Hawaii in general. But West Hawaii wasn't affected as much in 2014. But 2015, we had significant adult mortality. So seeing one of the sites starting at around 2015 start to increase is very interesting to us.
Nathan,
Yes.
does each dot include all the species?
Yes. So this is So for these graphs here, it is all species recorded at the site across all tiles. A little bit later on, there will be some looking at the 3 different genera and how they differ. So looking at the settlement, some strange settlement events, we have 2009 was abnormally high, and it was driven by 3 sites. You can see, so right here we have Kaupalehu and Puako up top, and then we have Honolulu, or excuse me, not Honolulu, Kalahiki of Ho'okena also had abnormally high those years. So you can see that the whole coastline is not necessarily experiencing these large events, and not even in the same region, right? So we have sites that are 100 miles apart, and they're also experiencing high levels.
They're at either ends of the island.
Exactly. And so for some of these, the questions is if it is regional, if they're all in an area and there's something happening in that area, but when it's not consistent, it's really hard to try to figure out what caused them. 2014, again, strange year. This one was driven by multiple sites, but none in the north. So the two sites with the highest settlement. So the sites that tend to get the most babies, Waikilo and Puako, they did not have high recruitment this year, while Honokahau, Ke'e, Ho'okena, Milili'i, and Manuka all showed higher levels. And if we look at the next year, relatively the same coastwide, right? But primarily driven by Puako and primarily driven by Porites at Puako. So now, is it significant? So these are the results of our GLM, our model. So if we look, do we have any site level significance? Like Helio and Puako are significantly higher than the other sites. So those 2 sites have many more settlers that come in every year compared to the rest of the coast. And then we have Port Ke'o, which has many fewer. And now, when we look at the change in settlement per site over time, we only have 1 site that's having a significant change over time, and that's Ke'o. And Ke'o is having a positive increase. So one of our sites, which has a lower amount of recruitment, is starting to have increases in settlement. And as we saw before, that's following a significant bleaching event. Very close though, depending on the year, Waikilio, one of our highest settlement sites, has a significant trend this year. When I ran it in 2020, using the same analysis, it was a significantly declining. This year, because it's not smaller than 0.05, it means that it is not significant this year. But as we said, with the episodic nature, with stuff going up and down, next year it could be significant once again.
Seems like there are a lot of undefined variables, more than you could count. Like,
Oh yeah,
rainfall, moonless nights, or full moons.
yes. And you have, yes, there's a lot of physical and biological aspects that go into the distribution and success of settlement, but it's really difficult with the episodic nature when the data keeps going up and down, and that's considered normal. It's really hard to have these environmental variables be highlighted as significant indicators for what's happening.
You need AI to analyze that.
Absolutely.
Larger data sets.
Yep, and exactly. So that's one of the things where it's like, now that was a perfect transition. So our power analysis. So essentially what we're looking at here is looking at our data based on what we've seen. How many years does a study like this need in order to start revealing significant data? And it's about 7 years. So what it means is certain settlement projects and looking at early life histories of corals, it's really difficult to really glean information in such a short period of time of 2-4 years. These studies really need to be long-term, 7-10 years, which is difficult, especially when you're trying to, find funding, staffing, and everything. And so continuity is a really important thing for these. So a little other information. So looking at the percentage, like how many of each of the genera are we finding? So about 50% of the corals that settle, we recorded, were of the genus Porites. 23% were Montipora, 13% were Polypora. And then past that, we start to get, there's Fungia. So the individual polyps, but they get really big. Have you ever seen, a lot of times they're sort of like tucked underneath, and it looks like one giant sort of coral polyp, because it's just one big giant coral polyp. And we do record them, we do identify them, but because they're not a significant reef building coral, they're not a priority for our analysis. All right, so we're looking at the three main taxa, so Polypora, Porites, and Montipora. Now, these are all tiles averaged per site, and then all sites averaged per year. So this is what we're looking like. This is what we're looking for as a coast. We can see that Porites settlement is higher, and Polypora and Montipora settlement is relatively consistent and lower. And that negative trend that we see in Porites settlement is significant. So across time, we are seeing a decrease in Porites settlement. But we're also seeing, so these points here are all Porites settlement events that are much higher than the average. So we're seeing very episodic Porites settlement. And then we have one little guy here, one little Montipora point, which is helping. That was 2009. So in 2009, Montipora had a good year. And we didn't necessarily see any changes in the amount of Montipora we saw around the coast. But that comes into play a little bit later. So we look here. We don't really have time to dig into this one too much, but this is the differences in the three main taxa at each site. And you can see there are some significant trends. So we can see that there's decline in Porites at Waiakilio, Honikahau, and Mililii. And some gentler trends, Manuka seen as well. But also some places are seeing increases in Montipora and Honikahau in the National Historic Park there. All right, now this is very busy, and I apologize. So what this is this is a list of different settlement studies that were conducted around the world. And what they're looking at is right here is sort of important to us. And this is their settlement densities that they found. So all of these studies done around the world, West Hawaii is right down here. So in comparison to a lot of these different studies around the world, we're having much lower amounts of settlement. So we have far fewer larvae that are coming to the reef and settling. All of these boxes in red are Hawaiian studies. And again, even with the different Hawaiian studies, we're a little bit lower. So there's some places that they found, like Hanalei Bay, just had choke settlement. It's very busy. It's fantastic. But some of us down here, we're down at 22, 8, 7. So in relation to other islands, we're also seeing that West Hawaii is having lower settlement than other islands. Yes?
Can you tell me anything about the different methodologies on the other sides? It
So
seems unlikely that Kauai would have.
So Methodology, there's a lot that use similar methodology. So the Kauai study is likely to have similar methodology because of the authors conducting that survey is one of the authors that helped design and develop this one as well. So there are potential that you can have based on where the tiles are, there can be that fine scale difference where you have tiles that are in an area which is, you have the proper amount of coarse rugosity so that there's a little dead space behind rocks. So it's really nice for them to come by and settle. And there can just be these areas that are really good for settlements. And then we have sites that aren't as good. So there is that little bit of variability in really how much we can directly compare them to one another. So yes, it is a little tough, but a lot of them are using the same terracotta tile. A lot of them are using the same array. There really isn't, they're using the horizontal with the gap underneath. So settlement tiles are fairly similar. So that experiment could have been run the same year in a slightly different spot, and the numbers could have been lower.
So related question, is your tile placement random?
Yes.
I mean, truly random?
Yes, so the tiles are randomized at a site. So they're based off of the DAR websites, and within that site, they are randomized based on heading and kick cycle using a random number generator. And so if any of the arrays break and there's no longer a place within a meter for us to resecure it, we have to re-randomize.
Do you have any, sorry, I keep asking questions,
Go ahead.
a whole bunch.
Yeah.
Do you have any information about the distribution within a cluster of 8?
Here, no. If we can chat after, and I can work up some stuff and send it to you if you like it. Yeah. But yeah, so right here, big takeaway from this is that West Hawaii in general, related to studies conducted around the world and also in areas of the Hawaiian archipelago, has relatively lower settlement. So one of the sites that we like to look at is Puako. One of the reasons is it has the highest settlement in our whole study. So Puako gets a lot of little babies in every year, and has had a few very large events. So 4 times, 5 times their average. And so theoretically, if we follow it for enough time and we're able to look at what's happening in adult coral coverage at the site, if you have a big event and all of them are surviving, theoretically you should see a bump or a boost in that adult coral cover, right? New babies come in, help to replenish the old ones and increase it. But that is not necessarily the case. So left-hand side, you're looking at percent community composition, and that's going to be your blue and your gold lines. So blue is your percent algal cover, and that gold is percent coral cover. Now, that is not necessarily the trend we like to see. You can see over the years, 2003, 2004, up until about 2020, our site at Puako has shifted from coral dominated to algae dominated. So theoretically, that means less space. Algae is going to grow over a lot of the available habitat that corals would like to settle on. And so having algae dominated systems are not the best for being able to replenish settlement. And so we have our 2 points here, and theoretically, you would be able to see a little bump in adult coral cover thanks to these big settlements in the years to come. But that did not happen, and that continues to decline. So using Puako as a case study and looking at that site, even with relatively high settlement densities, so even with getting a lot of babies every year, adult coral cover is declining. So there's possible that larval supply and available settlement density or settlement densities, they're not able to replenish, or they're not able to recruit into these communities in order to balance out the amount of mortality that's occurring, or be able to increase the amount of coral cover. So, and looking at this, even with high coral settlement, it doesn't look like those settlers are recruiting and surviving long enough to be able to join these colonies. And so what that indicates to us is that there's needs, like we need to mitigate sources of adult coral mortality. So what does it all mean? What did we find out with this study? So settlement is episodic, which means year to year, we can get high values, we can get low values, and that's all in the range of normal and expected. It also means that we can't depend on high settlement events in order to replenish declining adult coral colonies or coral populations. It also means that West Hawaii is likely recruit limited. So reasons for this is general geographic isolation, right? We're in the middle of nowhere. We have far fewer reefs to be able to supply larvae to us than some places like in Indonesia or the Philippines, where there's so many different sources where they could come from. We're really isolated here. So we really have to rely on the reefs on our own islands to reseed everything. There's a prevailing archipelagic current. So the general current of the Hawaiian archipelago is to go from southeast up to northwest, which means that we're down here on the very tip of that, and everything is generally going up and away from us. So things that are protected up north, while we may get individuals and things can come from the northwest down to us, in general, the northwest is not going to be providing us larvae. So protection up there and good reefs up there doesn't necessarily benefit us down here. So that means, okay, so we're more reliant on our own natal reefs. And also, so algal populations are increasing. So because algal populations are increasing, it means that there's less available habitat for settlers to start to settle in. So even if these larvae can survive that scary pelagic area and they make it all the way back and the currents are favorable enough to deliver them to where they need to go, if there isn't a good habitat for them to settle, they will either not be able to settle or they'll settle in a place that isn't favorable and they'll likely die. And this distribution is affected. We have seasonal, like big seasonal eddies and gyres, which on a very large scale, so 50 to 150 kilometers. And in the northwest, they tend to be favorable for settlement. So they rotate outwards and they deliver the larvae back to us. In the southwest, they tend to pull things in a little bit more. And if we get eddies pulling things away from the island, it means that there's a potential that they won't be delivered back to the island in time for them to be able to find food and for them to be able to settle. So we have fine scale eddies too. Anyone who's, if you guys have been sort of out on boats and things, you've seen the way that sometimes, especially when you get those current lines, and you can see when it's a spawning day or you get those algae blooms in there, you can see them start to spin and move around. There are times when we found cameras and they lost it 2 days ago and the camera stayed roughly in the same place. And there's also times where we found a camera and they're like, we've lost this 50 miles this way. So those fine scale, especially relatively shallow water, so once it starts hitting the shelf, how the water moves year to year and over the seasons, it's really hard for us to know that and to be able to predict that. So because we're likely recruit limited, recovery, if possible, is going to be slow. So since we sometimes have lava flows, it's a great opportunity for people to study settlement on lava flows because it's pretty much just a blank slate and they get to watch it over years. So older study, they looked and they estimated it can be up to 50 years for an area to reestablish after large mortality events. So what it means is it's going to take time and there's not going to be a quick fix. And so reducing recruitment bottleneck, so reducing the limit on the amount of individuals that can grow up and become reproducing adults is going to be really important for coral recovery. But how? A few things we can do, sort of maintaining healthy herbivorous fish populations. So they can be selective fishing or having areas where we don't take, because these fish eat the algae and they open up a lot of really good habitat and space that these corals like to settle on. So if we can keep as much of the space available, it increases the amount of little coral babies that are going to be able to come by and at least get a shot at settling. Mitigating nutrient and sediment runoff, things that can either smother them or increase algal growth. Same idea, preventing the introduction of pollution, effluents, and then more research, trying to figure out how some of these really complex and complicated sort of fine scale physical oceanography, coral biology, how it all interacts and works together, and just trying to figure out how it works, how we can use that to our advantage to figure out how to apply that to coral restoration and coral management. So that's kind of where we are. So acknowledgements, there are a lot of people that developed and staffed this project before me. Ross Martin, who some of you may know, is the most recent one. He really helped mentor this project. Dr. Eric Brown, who for a number of years did national park work here, and then now he's in American Samoa. And he's been really helpful with identification and statistical analysis and emotional support. Christopher Teague and Kailea Amandale, they're the PIs for national parks and for DAR. And then all the DAR staff, some of you here today who have helped out. And so a lot of these names you may recognize: Steve Cotton, Carlos De Vella, Laura Jackson, Leah Smith, Megan Lamson, Lindsay Kramer, Camille Barnett, Linda Prescott, Cynthia Hankins, Maggie Smith, Stacia Marcoe, Ashley Wills, Neil Hugenboom, Shannon Shea, Allie Craig, Ashlyn Overly, and Dr. Bill Walsh. Sally Beavers from National Park, who was here for many years. And then Jamie Go for NOAA, who helped out with some oceanographic aspects of it. Yay, references and questions. Yes.
There were a couple of studies in Australia that found that broadcasting the sounds of a healthy reef would attract recruitment or settlement of larval fish. Ears, but has there been any indication that that or any other intervention would increase coral settlement?
I don't know. But something with corals is they have less agency than fish. Fish develop a lot more, and they're able to have a better ability to react to those cues, like being able to swim in certain directions. Corals are small and they're relatively weak. So they're a little bit more at the whim of where the ocean takes them. So I'd say that I'm not sure what kind of that type of cues, but there's certain aspects of like having areas where they get delivered to shallow reefs, and then areas where they're not going to be exposed to fast currents or large swells. That might be one of the benefits. But as far as like that type of like sound or auditory studies for corals, I don't know. I'd say that would be less likely, less effective for corals than fish.
Why do they become pelagic? Is that has to do with genetic diversity or?
It's a really effective way, just like trees with pollen, it's a really effective way at dispersing. And so, yeah, that's sort of the main way that it allows them to get more of their babies, more places.
But they're not repopulating their own home kind of.
So if your currents are right, there's the potential, and there's also, so brooders might not go as far. And if the currents in certain areas are right, then more of the individuals may return to that place. It might not necessarily be because they're trying to, but more because the ocean just works in a way that it brings them back when they're ready to turn into little coral polyps.
Are there any laws, like new laws or programs that you know of to minimize runoff?
I do not know specifically about new laws, but it's a continuous effort. And something that's really hard here is it's hard to track, like, it's hard to track things that are passing through water because a lot of it's subsurface. And so you start getting into the realms of the alkaline pools. And so it's really hard for us to, if it comes out in a certain spot, it's really hard to track it back to a specific area. So I would say that I don't have a specific example, but more of just a general idea of the less things we can have going straight into the water, the better. And if we can have them go through more filtering sources, then that would be better as well. But I don't have a specific example.
New law, cesspool conversion program.
I've worked in labs where invertebrates are stressed to get them to spawn and that sort of thing. Is that possibly what's happening with the varieties during those El Nino years?
Potentially. So there was, in the Maldives, there was a heating bleaching event, and it was hypothesized that large numbers of settlement following that was due to sort of a stress response of, we got to get it out now, and do our best. So it's potential. From this study and from the data we have, it'd be really hard to prove, but potentially yes. And there are other places that have observed and thought the same thing.
I was just gonna ask, have there been any genetic studies done of the population of coral and their imbalance to sort of see where sources and sinks are?
Not yet, but I would love that. That's something that I would really like. If we can start to figure out where a lot of our corals are coming from, if we have really, really productive sources, then that's a way that we can aim levels of protection. So we can sort of have the idea of the spillover with marine protected areas, where we have this areas where we protect highly productive and high fecund individuals. And if they're going to be high producing a lot of little babies, then make sure that we protect that. But that's going to come through figuring out how things are connected.
Yeah.
And one of the tricky parts with that is you start working with these dispersal models, and it's really hard to estimate three-dimensional space.
Yeah.
It's really nice for certain ones like montiporids. When they spawn, they create these masses and they go and they sit at the surface. And a lot of those models are surface-based. And so those ones, you're like, okay, I have a little bit better idea of where they may end up, but there are certain aspects of early life history that we don't know specifics about a lot of our species. Hopefully that's something that we can do as we start getting new labs and new facilities that are going to have corals and work with that. That'd be very cool, and I would like that. And I think that's one of the ways that we could work to the future, but it's also very intensive and expensive. So that would be a full-on thing.
I was just thinking about the fluid code data that you were talking about and wondering if, based on what you were saying about the increase in algal cover, is it possible that you were finding more coral settlement because those were areas that were open?
Potentially.
As
Yeah, I would say that for a few of those years, maybe. But because it's consistently higher than the rest of the sites, and Huaco itself doesn't have a negative trend, then there's some sort of either association or there's something going on there that even though we are getting settlement, and there is high algal cover, there's some sort of relationship there, which it'd be really interesting to explore it more.
Yeah.
Potentially putting out more tiles and putting tiles out for longer and seeing if tiles that were out there for longer, if they're getting overgrown and if that's leading to mortality.
So across the coral species, there's a big variation in the pelagic time frame. My understanding is that cauliflower corals are very short, less than a day.
Can be.
Is there anything in your data, can you use that to determine if current is a a factor by looking at the lifetime for different species?
For this site, or for this study, we can't look at species because we don't know which species are settling, unfortunately. Like, that'd be very good. And also, while we have like a general idea, a lot of the pelagic larval duration that we understand of these species is very broad. A lot of it is like, it can be 1 to 200 days. You're like, okay. But yeah, most of them are on the shorter end, but each aspect, when there's a little bit more wiggle room and there's a little bit larger of a range, when you start putting those together to try to answer a question, you start introducing more and more bias, so it becomes harder and harder. So with this study, we're not able to speak at the species level. I wish we could, but yeah.
It's a non-science question.
Okay.
It's very clear from your presentation, a great presentation for a non-scientist, that you really love your work. So I'm curious why you like these polyps. Hang on. And second to that, it's like a follow-up, based on 12 years, and we hope it's another 15 years, 4 years. What's making you optimistic?
Well, I'll start with what's making me optimistic is that we didn't automatically see a huge decline in settlement at both the coast and site level following 2014, 2015. So it means that it's more complex than that. But if we're not necessarily losing the amount of individuals that are coming in, then there are ways that we can sort of approach that. And as far as one of the reasons why I like doing it is looking at the tiles themselves, because when they're in the water.
That's science. Why do you like this animal?
Oh, this animal?
Yes.
Ooh, I would say it's very love-hate with this animal because they're like very cool and interesting, and the tiles themselves, there's a bunch of random stuff on it. So every time picking them up, there's like all sorts of weird, like little crabs and stuff, and like things you wouldn't normally see. But corals themselves are very fickle. It's very love-hate. They're very cool, but they're very frustrating to work with sometimes. Like with the data, because it's so. Huh? No, it's a joy every day. Like you saw from the data, it's so episodic that, you have high values, low values, high values, low values, and all these values can make it so it's very hard to have things to say, especially in settings like this, where you noticed how there were 4 or 5 sites that we didn't really touch on. And that's because it's really hard to say when the statistics don't allow you to. And so what you really have to reel yourself in, I think, when especially talking about settlement, because there's a lot of it that you can easily get in front of what your data can say.
If you allow me, it sounds as though it's a little engine that is doing.
Yeah, it's consistent and it's chugging away, and I think there's definitely things that we can do. We have a little agency in being able to try to help them along a little bit, trying to increase the amount that are able to find space to settle, doing things to mitigate things that would cause mortality in those individuals that are trying to become adults. So there are things that it gives you just enough hope to sort of dive in.
You're a dad.
Yeah, that's true. Yes.
Do you think Keoho could be so low because there's so many people and so many boats going in and out?
Same question.
I can't really answer that. There are a lot of possibilities, but unfortunately, it's really hard to. Those are potential factors that could be introducing things into the habitat that would negatively impact it. But with this study and what we have, it's really hard, and it would be. I can't really say if it is or not.
I have another comment. This has nothing to do with your coral study. But with what went on in Lanya. I'm wondering if anybody's going to be studying the ocean there and see the impact of that, because it seems like it's going to kill everything.
I'm sure that people are asking the same questions, and I'm sure when the time is right, they'll figure out, if appropriate, when to be able to sort of investigate that, because it is one of those things that if it is possible and appropriate, that the data could reveal some things that might help us.
Every answer reveals 1,000 questions.
Exactly.
It sounds like you need more parameters to determine correlations, and also some of this questions about why things are happening.
Yes.
Is that on the agenda going forward?
Yes, so it is, and we're able to get a big sort of environmental data set that came in from NOAA satellites and things like that. The first approach we took didn't really reveal much. We did non-metric multidimensional scaling, which essentially puts everything in a three-dimensional space. And when things cluster together, it means they're more related to one another. So if it's clustered, if a site is clustered towards some environmental factor, like water temperature, like, okay, the water temperature is influencing settlement at this site. And it did not reveal much. It was, we were able to get, whether we had 1 or 11 variables, we were only able to provide 10% explanation for the variability of settlement rates.
Eliminating parameters is just as important as finding the right ones.
And unfortunately, like with that, it's really hard to, like, we're not sure we can eliminate them. And so the next sort of approach is figuring out different ways to try to fit all of these blocks together to see if we can say, like, no, these have nothing to do with each other, or if it's like they fit together just enough that we want to investigate more. Yeah, and it's all one of those, like, all right, you write the code, you figure it out, you learn the method, and then you hit a brick wall, and then you just have to talk to people and figure out the next approach.
I think whatever the data tells us, I appreciate you going out and gathering. I think a reason to be optimistic is that 20, 30, 40 years ago, I didn't see anybody from DAR. I mean, it didn't exist, going out and studying this stuff. So the very fact that now you're trying to save those corals, or even though they're jerks sometimes, I appreciate it. And I think if anybody can do it, you will.
Thank you, Jeff. Can you come with me to all my presentations?
No ringers.
Yeah.
Is there any research that's been done that looks at survival rates, like past the settlement?
There are, and that's something we can get into if you want to, like, chat more about that later. Yeah, it's different design, and so there's ones where they'll have tiles and they'll keep them wet and look at them and replace. We're not able to do that with my site because they go to a farm upstate every time we collect the tiles. But yeah, so different tiles, different, like, study design, and yeah.
People have done that.
People are looking into it. Again, it's one of those things where it's like it's like long term, and it's really easy for some projects like that to get shelved. Just if you have the right amount of turnover, or if you have the right, different aspects here and there, it's really hard to have those continue. Yeah.
Other than having fish eat the algae, are there any other ways to, like, proactively reduce the algae in these areas?
Ooh, that's a good question. So so in areas where there aren't too many urchins and other herbivores are one of those things. And another thing is having healthy coral assemblages. Like, algae is present, and that's okay. But it's when you have, you'll have a coral head, and if it dies, and then that space is no longer taken up by a coral, and then competition starts. And if the environment is more conducive to, like, if there's a certain amount of nutrients, like you have some nice runoff, and the right amount of sun, the right amount of water temperature, and the right amount of nutrients, algae gets very excited and very happy, and it grows really, really fast. And allows them to outcompete the different things. So, like, chemical oceanography, so, like, a lot of the water quality and some things like that can be a way. So say, yes, there are. Herbivores is one of those things that we can, I would say, more easily act upon. So that's one reason why it comes up a lot, because it's one of those things where we can see it. We're like, okay, this is something we have. This is something that we can work on, and it's easy to communicate.
On that water quality front, has anyone explored kind of connecting like coral health to water quality standards so that you could actually start enforcing things where, water quality standards violated if it's impacting coral health?
Man, I wish Lindsay was still here, because I would have just punted that over to her real hard. Yes, there are people looking into that. I'm not too familiar with it. But if you give me information, then I can connect you with people who can answer your questions. Because that's one of those sort of, we go down to this project and we go very fine. And that's like scaling back out.
Yeah.
It's like all the different groups and organizations working on different projects. How can we all sort of share information that way so that we can, because that's the way that we can lead up to like good management and like good decisions. All right. Well, thank you very much, everyone, for coming out. I appreciate it. All right.
Well, thank you, everyone.